Okay, so as Joanna mentioned, I did my PhD in Robert Prividel's lab, where I developed three-photon microscopy with adaptive optics, where we can really achieve high-resolution imaging deep inside the brain. And our group develops optical tools that allow us to study the brain and to understand the function of the brain, how it is connected, the architecture, and how it processes information has been of great interest to scientists and society over centuries and decades. However, in most situations, we cannot get access to the human brain, and therefore the mouse as a mammalian organism has become a widely used model organism in neuroscience. So as you probably know, ideally we would like to have an imaging technique available which allows us to study the entire mouse brain at synaptic resolution and at volume rates comparable to action potential. However, the mouse brain is still too large and additionally highly opaque and scattering. And therefore, we do not have one imaging technique available which allows to cover all of the required parameter space in terms of imaging speed, field of view, and at large depths and high resolution, but we rather have specialized microscopes which target different areas of the parameter space. And the aim of my PhD was to push microscopy with respect to achievable imaging depth and resolution while imaging the in vivo mouse brain. And multiphoton microscopy has become the gold standard imaging technique when imaging inside scattering tissue such as the mouse brain. And in multiphoton excitation, a fluorescent molecule is excited with 2 or 3 photons of double or triple the wavelengths compared to its 1-photon excitation. And due to this higher order excitation, the photon density of the excitation photons is only at the focus is high enough that fluorescence is generated, which leads to inherent optical sectioning. And this comes with the advantage that both scattered as well as ballistic photons now can be detected, as their origin can be assigned back to a single point inside the volume, therefore significantly increasing the detection efficiency. And additionally, nonlinear excitation allows to shift the excitation more to the near-infrared spectral range where scattering is reduced, and hence penetration into the tissue also enhanced. But also with a good 2-photon microscope, we can currently only investigate the upper layers of the cortex at high contrast. Although 2-photon microscopy can be pushed towards a millimeter, however, with the trade-off of having reduced contrast at depth due to the onset of out-of-focus fluorescence at the surface with increasing excitation power at depth, which you can see in this image here that the large bright structures such as the somata are still visible, but contrast to resolve fine structures is lost. However, now if we use 3-photon excitation, one can image much deeper into the brain due to even further long wavelengths excitation, and 3-photon microscopy was pioneered by the Chris Chu group and first demonstrated in 2013. And as you can appreciate here in this 3-photon image acquired at the same depth, now really the fine structures can be resolved due to the improved contrast. And this fundamental improvement in signal to background ratio in 3-photon microscopy results from a later onset of out-of-focus fluorescence due to the higher order nonlinearity. But a second consideration we have in microscopy is image quality degradation due to optical aberrations. So in the ideal world, we would have the same refractive index between all materials, leading to a perfect diffraction limited focus. However, in the real world, we do have refractive index mismatches, which lead to deformation of the wavefront and therefore also this degradation of the point split function and resolution. However, the accumulation of aberrations is an entirely deterministic process, and therefore an ideal modulated input wavefront can be found, which allows to recover for all aberrations, and therefore near diffraction limited resolution can be restored. So in order to enable really deep imaging and high resolution, we use 3-photon microscopy to increase the imaging depth compared to conventional 2-photon excitation due to a longer wavelength excitation and a fundamental improvement in signal to background ratio. We use adaptive optics, a technique which allows to correct for wavefront aberrations, and therefore we can recover near diffraction limited resolution deep inside the brain. And as we're imaging the in vivo mouse brain, we also implemented intravital imaging strategies, which allow us to correct for motion artifacts resulting from vital functions such as the heartbeat. And we also had a special focus on the light exposure to maintain physiological imaging conditions. So here's a schematic of the experimental setup. To enable three-photon excitation, which is a highly unlikely process to occur, we use a very low repetition rate, high peak intensity, short pulsed laser sources. And as we are working with the short sub-100 femtosecond pulses, dispersion compensation becomes essential, as the three-photon signal is inversely proportional to the square of the pulse duration. And we are currently using a pulse compressor based on a single prism and two roof mirrors, which allows fairly fast tuning of the dispersion. And I've also implemented an adaptive optics module, which allows us to correct for wavefront distortions, and I will discuss this later throughout the talk. So which new biological questions or structures can now be addressed with three-photon adaptive optics microscopy? So three-photon excitation allows to investigate subcortical brain regions such as the hippocampus non-invasively, which previously required invasive procedures such as the removal of overlying brain tissue in combination with two-photon microscopy. And recovery of near diffraction limited resolution with adaptive optics allows to visualize small structures such as spines at larger depth, but also the recovery of more efficient fluorescent excitation can also be essential for when one is interested to study really heat or photosensitive processes, as these allows to also reduce the excitation power while maintaining the same signal level. And to establish adaptive optics correction and to evaluate the correction efficiency in vivo, we needed a resolution target deep inside the brain. And spines are an ideal resolution target, which are small structures of submicron size. So for our experiments, we're using a Taiwan GFP mouse line, which has pyramidal or excitatory neurons labeled with GFP. And to obtain optical access to the brain, we use a cranial window preparation, and surgeries are done by Juan, a postdoc in our lab, where we remove a part of the skull and replace it by a cover slip. And here I would like to show you some of the results with three-photon microscopy at 1300 nanometer excitation wavelength in the visual cortex and hippocampus. And here in blue, you can see the third harmonic signal, which mainly visualizes blood vessels. And in green, you can see the neurons, which are labeled with GFP. So with a good two-photon microscope, we would currently achieve high contrast CRISPR images approximately down to 600 micrometer depth. However, now with three-photon excitation, one can image much deeper into the brain, down to 1.2 millimeter, where here you can appreciate that we can clearly visualize the vasculature throughout the layers in the hippocampus. And here you can appreciate that we can clearly resolve all the neurons in the entire cortex. We can clearly see the fibers of the corpus callosum, which are the long-ranging axonal fiber bundles connecting the two brain hemispheres. And we can clearly see the CA1 hippocampal neurons. And if we now have a look at the cross-section acquired at layer 6 at around 800 micrometer depth, you can also appreciate that we can clearly resolve all the fine details, so the dendritic branches. And if we go deeper, we can clearly also see the fibers of the corpus callosum. And if we go even deeper at beyond 1 millimeter depth, we can see the somata of the CA1 hippocampal neurons. And if we change the contrast, you can also appreciate that here we are still capable to resolve the fine structures, such as the dendritic branches at over 1 millimeter depth. But remember, the goal was to have synaptic resolution, so really high resolution throughout an entire cortical column. And if we have a look at the zoomed in image here at layer 6 at around 800 micrometer depth, you can still see that it's rather difficult whether there are actually spines on this dendrite or not. And that is why I've implemented also two complementary approaches, intravital imaging and adaptive optics, to enhance the visibility of very fine dim structures deep inside the brain. And one of the challenges we are facing when imaging the in vivo mouse brain is tissue motion on the micron scale due to the heartbeat, which you can see on this video here, where you can see that we really have some significant wobbling of the structure. So one standard technique to improve the signal to noise ratio of fine dim structures such as the spine is to perform frame averaging. However, if we now perform frame averaging with these consecutively acquired frames with high content variability, what we end up with is a complete blurred image where it becomes even more difficult to resolve the fine spines. And that is why we implemented the capability into our microscope to gate our image acquisition in real time to the cardiac cycle. And real-time data processing of the ECG signal is performed by an FPGA, which was programmed by Matteo and Ling from our group. So we use now the microscope clock to gate our image acquisition in real time, so that acquisition is only performed in between the heartbeats where tissue motion is minimal. And here you can see another example of a neuron acquired at 700 micrometer depth, where here you can also see without cardiac gating, we really have this high frequency motion artifacts, this wiggling, which disappears if we enable cardiac gating and we rather persist with this low order distortion and translational movement. And another way to visualize the motion artifacts is via standard deviation projection images, where also here you can see that we have high content variability at the edges due to the wiggling of the structure from the heartbeat, which can be removed with cardiac gating. And another way to compare the frame-to-frame similarity is to perform cross-correlation of individual frames to one another. And also here you can appreciate that we have a much higher content stability if we enable cardiac gating. So cardiac gating allows us to improve the signal to noise ratio of fine dim structures by performing frame averaging, but it does not allow us to improve the real resolution of the microscope, which can be done with adaptive optics. And in adaptive optics, we differentiate between two main type of aberrations, mainly system aberration, which result from the optics themselves and due to the sample preparation, so the refractive index mismatch between the objective, the immersion media, and the sample. And then we have the sample aberrations, which result from the tissue itself. And while a correction for system aberration yields optimal performance of the microscope, sample aberrations can only be corrected with an active optical element, such as a deformable mirror, which can modulate the wavefront of the light. And we are using an indirect wavefront, modal-based wavefront sensing approach, where we use a continuous membrane deformable mirror, which we drive with Zernike polynomials. So we drive the entire mirror shape at once. And to optimize the mirror shape, we use an iterative approach where we use the fluorescent signal intensity as a feedback signal to optimize the shape of the mirror. And to give you a feeling of the acquisition or correction parameters, so generally we use between 20 to 40 modes. And we treat the modes orthogonal so that they can be optimized independently, which means that 5 to 9 amplitude REMs need to be performed per mode so that the optimum per each mode can be defined. And this can be acquired within 500 millisecond acquisition time compared to the overall correction time, which is over 30 seconds. And this independent treatment is essential, especially for in vivo biology, where we can have fluctuations of the fluorescent signal due to motion or due to bleaching, or even due to intrinsic fluctuation, which is especially important if we consider functional imaging. And here on the left, you can see how a normal optimization procedure would evolve over the correction time. So you can see the signal intensity plotted, and we can appreciate that we get a clear enhancement of the signal intensity during the first iteration and only a minor improvement in the second and the third iteration, which indicates that our assumption that the modes can be treated and optimized independently is valid. And now I would like to show you some of the results we achieve with adaptive optics correction in vivo and three-photon excitation at 800 micrometer depth at layer 6. And here you can appreciate that with full aberration correction, we achieve a clear enhancement in both intensity as well as the resolution, where the fine structures such as the spines only become clearly visible after aberration correction. And you can also see on this intensity plot here where the second spine only became visible after aberration correction. And if we now have a look at an orthogonal view across a dendrite, you can see that before aberration correction, the point split function is severely distorted, especially in the axial dimension. While with full aberration correction, we are able to recover the resolution, which leads to a 6-fold increase in intensity and a 2-fold increase in resolution. And here you can see another example also acquired at 800 micrometer depth at layer 6, where you can also appreciate that we can now, with full aberration correction, resolve all the fine details so that we achieve synaptic resolution really throughout an entire cortical column. You can also see in this video here where you appreciate that we really can see all this fine detail. But we can also perform aberration correction at over 1 millimeter depth in the hippocampus. And here you can see another example where you can also appreciate that with full aberration correction, we can really resolve all the fine details at dendritic branches, which were not visible before aberration correction. So here we achieve an intensity improvement on large structures such as the somata, which is 3-fold, because large structures are less affected by aberrations when the point split function becomes degraded, while smaller structures such as the dendrite, we achieve an intensity improvement of over 8-fold, as these structures, fine structures, are more affected by aberrations. And we can also quantify the improvement in resolution in the lateral dimension in a more quantitative way by a Fourier transform analysis. And also here you can appreciate that both in the cortex as well as in the hippocampus, we get a clear enhancement of the high spatial frequency component after aberration correction, which leads to an improvement in lateral resolution. And we can also analyze the improvement in axial resolution via intensity plots along small structures such as dendrites. And here you can see the axial full width of max plotted for different depths. And in blue, you can see the point split function, the axial dimension before aberration correction, where you can clearly see the trend that the point split function degrades with depth. While in all cases, we're able to recover near diffraction limited resolution with adaptive optics. So now that we achieve high resolution near diffraction limited imaging and three-photon microscopy at over 1 millimeter depth, we set out to also study a different cell type in the brain, glia. And in this project, we worked together with the group of Amit Agarwal at Heidelberg University, who is specifically interested in astrocytes. So astrocytes exhibit a multitude of functions, and these star-like shaped cells branch out into the tissue, and they form thousands of contacts with synapses. However, unlike neurons, astrocytes are not electrically excitable, but they do exhibit calcium transients, which you can see in this video here, where the astrocytes are labeled with a calcium-sensitive probe, GCAMP. And this calcium transient, they can vary both in space and in time, but also in shape and in size. And a subgroup of this calcium transients, which are associated to synaptic activity, so-called microdomains, are actually fast enough to potentially play an active modulatory role. However, the functional significance of these microdomains and whether astrocytes participate in real-time decoding of information is still not well understood and is currently under investigation. So most of the knowledge we have of astrocyte calcium signaling in vivo is obtained via two-photon microscopy in the upper layers of the cortex. However, like neurons, there are different classes of astrocytes, mainly protoplasmic astrocytes, which reside in the gray matter, so the cortex, and have already been investigated in vivo with two-photon microscopy. We also have fibrous astrocytes, which reside in the white matter, so the corpus callosum. And due to their deep location in the brain and their location amongst the highly scattering fibers of the corpus callosum, have previously not been investigated in vivo. So that's why we set out with our three-photon microscope to study both cell types, protoplasmic as well as fibrous astrocytes. And here you can see an example of a protoplasmic astrocyte recorded at layer 6 at 800 micrometer depth. And here the astrocyte is also labeled with GChem. And here on the left, you can on the right, you can see the automatic detection of the calcium transients of the microdomains, which is performed via an analysis pipeline called Cascade, which is developed in the lab of Amit Agarwal. And data analysis here was performed by Khalil, a master student in his lab. And in this example, you can appreciate that we were able to detect 90 individual microdomains with a good signal-to-noise ratio. And if we now even go deeper into the brain, we were also able to record calcium transients from fibrous astrocytes, which you can see in this video here. And to the best of our knowledge, that's the first in vivo non-invasive recording of these cell types. And also in this example, we were able to detect 58 individual microdomains, also still with a fairly good signal-to-noise ratio. But adaptive optics and aberration correction can also be essential when recording from astrocytes, as astrocytes are extremely light and heat sensitive cells, which quickly change their physiology upon prolonged light exposure. So recovery of near diffraction limited resolution with adaptive optics can be essential in two ways. First of all, higher resolution allows to capture more finer microdomains, which are of high interest. But secondly, recovery of efficient fluorescent excitation also allows to reduce the excitation power while maintaining the same signal level, which enables more physiological imaging conditions for the astrocytes. So here you can see an example of an astrocyte recorded first without adaptive optics, where you can see that it's rather difficult to tell whether there are calcium signals occurring. But now we enable adaptive optics correction. Now we can really appreciate that we can visualize the calcium transients traveling through the astrocyte. So adaptive optics correction leads to a clear enhancement of the signal amplitude. But more importantly, adaptive optics correction enabled us to achieve a 6-fold increase in the detection efficiency of signaling events. So before aberration correction, we could detect 9 individual events, while after aberration correction, we were able to detect 56 individual events. So in summary, I've presented you three-photon microscopy in combination with adaptive optics, which now allows us to recover near diffraction limited resolution down to a millimeter depth in the brain, which now we can address new questions previously not addressable. For example, we could now study synaptic remodeling in a layer-specific manner. And we can also now investigate the differences in the calcium signaling between protoplasmic and a previously understudied fibrous astrocytes. But the microscope can not only be applied to mouse neurobiology, but can also be applied to different model organisms or different biological fields. And with this, I'm at the end of my talk, and I would like to thank all the people involved in this work, the people who contributed to the work I presented today, I highlighted throughout the talk. And yes, I would like to thank all the members of my group and my collaborators, and I would like to thank you for your attention, and I'm happy to discuss a bit more and answer your questions. And as I learned that we still have a bit more time, we can also go a bit more into the technical details if you're interested. Thank you.
Thank you, Lina. Super fascinating work, very, very interesting. I'm sure there's plenty of questions from the audience. I see already a raised hand. Mariana, do you just want to unmute yourself and ask your question?
Yeah, I'm sorry. I don't know if you spoke about it, but I had a connection problem at some point. What kind of adaptive optics are you using? Is it like a crystal, reflective adaptive optics? Because I know it introduces a lot of losses in in the system. And the second point would be, do you correct for aberrations interactively, or is you use a specific pattern that, because it's kind of tricky to get these corrections done in in a specific way. I know that each individual has a different correction.
Yes. Yeah, thanks for your question. So we're using a reflective, so we use a deformable mirror, and hence the losses are negligible. It's like any other mirror which you insert into the setup. And we choose a deformable mirror. One advantage is that, I mean, the SLMs become faster, but still the deformable mirrors are faster to be controlled, and they are wavelength dependent. SLMs are always polarization and wavelength dependent. So if you are not working at the same wavelengths, it's more recommendable to use a reflective mirror-based approach. So that's the first part. And the second part is, so we are doing the correction mainly for on the astrocyte or on the neuron somata. And one of the advantages in multiphoton microscopy or three-photon microscopy is that the fluorescent signal depends non-linear on the excitation power. So your signal is quite, so if you shrink down your point square function, you get an improvement, you get a larger signal improvement. So you're quite sensitive towards that. What is true that it is quite sample dependent. So one has to be really careful that, for example, you characterize your deformable mirror really well, that especially when you use the continuous membrane deformable mirrors, because they are nonlinear devices, so the modes can couple between one another. So for example, we correct our deformable mirror very, very carefully, that if, for example, we apply a spherical mode, often in the initial calibration, you also get a defocus shift, which you really don't want to have, right? Because then you could just have the intensity fluctuating because you're shifting around in your sample. So we really carefully characterize it that we stay in the plane where we want to. And then we do the correction for the intensity. But these are a few things one, especially for the calibration, one has to do that very, very carefully. Yeah.
Quite tricky, but it's nice.
Okay, Frauke, do you want to ask your question next?
Yeah, thanks. So thanks for that really fascinating talk. Great work and congratulations. Maybe you could elaborate a little bit more on how you prepare the mice for the imaging sessions. And you mentioned that this is basically non-invasive. So I was wondering if you could also do longitudinal studies and, for example, establish a training paradigm.
So non-invasive, so we are still using a cranial window preparation. However, that's accepted in the community that if you wait for a month for the inflammation to come down, that this can be considered non-invasive. And when we reach the hippocampus, for example, people normally remove then the overlying brain tissue, and this we don't do. So hence we can record, depending then on the window quality, we can record the same mouse over 6 months. So what this microscope currently, we don't have an awake station, so there you kind of need extra, for example, that the mouse needs to be on a wheel. So then if it is awake, if it moves, you somehow need to damp that movement and reduce also the stress for the animal. So as we are not really doing any behavioral studies yet, the mouse or all the data I showed were with anesthetized animals. Okay. But that could in principle be adapted, and we have another microscope based on temporal focusing for really fast high volume rate imaging, which are done on awake recording. With the behavioral studies, so one of the, let's say, intrinsic disadvantages of three-photon excitation is that we have low repetition rate laser sources, because you really need to the absorption cross section is orders of magnitude smaller for 3-photon excitation than it is for 2-photon excitation. So you need to compensate somehow for that. So you really need pulses with high peak intensity to have a very high photon density, but you have also this constraint by biology that you cannot have too much average heat in the tissue, because then you just heat up the tissue. So what we now do is that you increase the peak intensity, but you lower the rep rate of the laser to still have the same average power. So we are currently working with a 400 kilohertz laser, while normal 2-photon imaging is done with 80 megahertz. So we're 200 times slower. So that's an inherent disadvantage if you really push for the depth, that you're slower. So you're restricted to smaller field of views, basically.
Okay, thanks.
Mm-hmm.
Actually, can I ask a direct follow-up question on this? So you've shown very nicely the calcium imaging. Do you think it's possible to also go towards imaging with membrane potential, so even faster imaging at these steps?
Sure. I mean, in the end, it depends on the field of view. You have we have as we are not using cameras, this point scanning. So we are really limited by the repetition rate. We have 1 pulse per pixel, and then you can basically do the calculation when do you need to have to be back in your frame. So how many pixels can you actually acquire within that given time? Which for voltage imaging will not be many.
Yeah. Okay. Thank you. That's very interesting. We have a question in the chat that I'm just going to read out from Maria Cockley from Spain. And she asks, I would like to know whether you have looked or plan to look at the perisynaptic processes and or the end feed of the astrocytes, for example, with regards to movement of the processes, calcium transients, in response to neuronal activity or changes of blood flow.
No, we haven't done that yet, but we could potentially do this. So we now that we have established also the parameters to record astrocyte calcium transients, and we also still have the collaboration with Amit Agarwal's lab going, that we will address now different questions in the field of glia biology. Especially, we're interested in the changes of the morphology and the corpus callosum, for example, during demyelination. These are things we are currently, or in the future, we'll probably investigate, but haven't looked at it yet, so I cannot answer or give you more insight until now.
Thank you. I think there's a lot of interesting things that can be explored with these technologies.
Yes.
John, do you also have a question you want to ask?
Thanks, Lina. Excellent presentation, very interesting.
Thanks.
I was just thinking I guess the instrumentation that you've been using, it's largely custom made, right?
Yes.
So then how far are we in terms of the turnkey instruments that are available to reach this kind of corrections that you're talking about?
So 3-photon excitation, I think there are already some custom lasers are ready to be turnkey. However, adaptive optics still requires quite some expertise. So we are not there yet that we have an image metric available which would work for every sample. So that still needs to be optimized and needs some expertise tweak. And there are groups working on a more universal framework, and as we in multiphoton or 3-photon excitation, we only correct for the aberrations in the excitation path. And as I mentioned, we have this really high nonlinear dependence of the fluorescent signal on the excitation power. This is quite robust, but I think still needs a bit more optimization to be turnkey. But hopefully we get there.
Thanks. I was also curious with applications. You showed a few very nice ones. How is it with the instrument that you have used, where you get very, very significant depth actually? Has there been any interest of using that for studying gliosis, which I guess would be quite a rewarding application area?
Not yet.
Okay.
No. So as we are so at the end of my PhD now, all the things come together. Now it's working really nicely. That will be the next step, exactly, to explore more biological fields.
I think those in brain damage would be potential customers of your instrument.
Mm-hmm. Yeah, I mean, it's great then to see that people actually find the technique useful and can use it to answer their questions.
Okay, thanks.
Mm-hmm.
Can we have a question from Julian?
Yeah, it's very simple. Maybe I missed that, but which multiphoton laser are you using?
So we're using the Spirit NOPA from Spectra Physics. So it's a pump laser at 16 watt at 1054 nanometers, and then this pumps a NOPA, non-collinear optical parametric amplifier, which is based on different frequency generations. So we are getting two beams, one more in the near infrared, so which we use for 3-photon excitation, which we can ramp between.
You have an OPO. You have an OPO, or you have it already on the multiphoton laser.
So we bought both from a company, so we didn't didn't custom build the NOPA. No.
Okay.
Mm-hmm.
And you don't have any pre-chirp or any. You don't need to do that. Pre-chirping your multiphoton.
Yes. So that's custom built a pulse compressor based on a
pulse compressor.
Yes, exactly.
Is that the prisms?
It has one prism and two roof mirrors.
Okay.
And that allows relatively fast tuning of the wavelengths and the dispersion. And this you certainly need. So without pulse compression, I think the pulses are around 500 femtoseconds long, and before they were like 50 femtoseconds long. So that reduces the signal 80-fold or more. So you really need to compensate for that.
Yeah, okay, it's good to know, because I mean, when you get so deep, I guess you have to really compensate. Sometimes people have the discussion about yes or no pulse compressor, but in our hands,
in 3-photon certainly you need it.
Yeah, okay.
Otherwise, you need to put too much heat into the brain.
You will have to increase the power quite a lot, and you say that you're already slow in some way, and then you will heat even more.
Exactly, you have more heating also in the more near infrared range, so you really want to optimize to minimize.
Yeah, that was the other question about the heating, because if you have a slowdown, how you see that? Because of course, if you go in the brain and you want to look, and for example, if you want to study lymph nodes and see the B and T cells, that we know by experience that the temperature is very important. I mean, in brain maybe the oxygen is more important, but in other cases the temperature is, and we have noticed by using multiphoton that sometimes we stop the cells moving because they overheat.
Yeah. So we haven't investigated that, but the group of Chris Chu, who kind of like pioneered 3-photon excitation, have investigated that. And for the brain, this also holds to be true. So currently the accepted value in the literature is 100 milliwatt down to 1 millimeter depth, and then you get close to, I think, 42 degrees at the surface, and much more you should not exceed. Exactly. So there are also limits. Otherwise, you heat up the sample too much, and you don't want to do that if you want to maintain physiology.
Yeah, well, you want to maintain. I mean, the thing if it's you heating too much, you don't know if these movements or things, it might be as well because of the heating. Yeah. Thanks.
Are there other questions? Anybody else want to ask something? You can just raise your hand or just unmute and ask your question directly if you want. Okay, if there's no more questions, then thank you again to Lina. Thank you for letting us ask all these questions to really dig into some of the technical details of the setup. I think this is really interesting for all of us to hear some of the technology details behind this amazing application. Okay, and then I think to wrap up this session, I just want to share with you that we have now published our spring schedule for the virtual pub. So up until the end of May, we have really exciting lineup of speakers covering a wide variety of topics, from image analysis to visualizing new tools to oblique plane microscopy. And so you can find the details of this on our website, and we really hope to have you join us for future events, especially for our special speed pub next week, as I already announced. And yeah, I think with that, we're going to wrap up a little bit early, but I'm sure everyone appreciates extra 15 minutes to get a late lunch or an early coffee break. And I wish you all a nice Friday afternoon and then a good weekend. Thank you, everybody.
Thank you. Bye-bye.
Bye-bye. Have a nice
Bye.
weekend.
Bye-bye.